All right, it's time to move on to chapter D3 on the liver. All right, well, in order to understand what the liver does to your blood, we got to understand where this blood is coming from. So let's do a super simplified drawing of your liver. Okay, so here's your liver. I put a real picture up here so that you can see a little bit better as to what that looks like. All right, so we've got two blood vessels taking blood into the liver. Okay, so both of these go that way. All right, this first one is called the hepatic artery. Now, HEPA, you're going to see this a whole ton in this chapter that refers to the liver. Artery meaning it's coming from the heart. So this guy is going to carry oxygenated blood from the heart to the liver. Okay, so the liver's an organ, it needs oxygen and glucose and all that good stuff too. All right, this other one down here is called the hepatic portal vein. Well, it helps if I spell that right. Hepatic portal vein. Okay, and so this guy is carrying blood to the heart. So it was somewhere else in the body, I don't know, in your leg, okay? And it needs to get back up here to your heart, but it's going to go through your liver first. This guy carries deoxygenated blood. Okay, so the liver is kind of unique in the fact that it's carrying both oxygenated and deoxygenated blood into the same organ. Now, some cool magical stuff is going to happen in the liver, and then there's one blood vessel leading out of the liver, and it's called the hepatic vein. Okay, so just a vein, just regular vein, okay, meaning it's carrying things towards the heart. Okay, so again, once things leave the liver, they're gonna run up here through the hepatic vein to your heart. And this guy's carrying deoxygenated blood, okay? Any blood or any oxygen that was in this blood has been consumed by the actual cells of the liver. Okay, now for all of you future surgeons out here, don't expect to cut someone open and see things in such a nice orderly arrangement here. In real life, that hepatic artery and portal vein enter down here, and this one goes up here. Okay, but just to make things easier, we're gonna draw it this way. All right, so let's discuss the two blood vessels that are carrying things towards the liver. So the hepatic portal vein is one of them, and it's carrying blood from the capillaries of the small intestine. So we talked a lot about digestion and about how inside the small intestine are these villi, these finger-like projections, right? And if I take a closer look at the villi, I have these arterioles coming in, okay, and then they're absorbing a bunch of nutrients, and then they leave through this venule. Where does this venule go? Well, it forms with other venules from other villi, and becomes veins, and eventually all the blood from the small intestine ends up here in the hepatic portal vein. So it's deoxygenated, right? All of the oxygen was consumed by these cells of the villus, so it doesn't have any oxygen in it. It's being carried by a vein, so we should know by now that that means low pressure. And depending on what's going on in the digestive process, it can either have a lot or a little nutrients. So if you just ate a big meal and there's a whole bunch of food in your small intestine, that food is going to get transported by these capillaries and then venules and then join in the hepatic portal vein, and we could have a whole lot of nutrients in there. If you haven't eaten in a while, probably low on nutrients, okay? So it varies depending on your time of diet, but for sure that sometimes it's going to have a whole lot of nutrients. All right, so this hepatic artery is quite a bit different. So we need to remember that the liver is made up of live cells and they need oxygen also. So we need a blood vessel that's gonna provide that oxygenated blood. That comes from this hepatic artery. So that's carrying blood from the heart like every other artery. It is oxygenated and it's at a high pressure. There is, however, low nutrient content. Okay, we're not bringing the blood in here because the liver needs nutrients. This blood is coming into the liver because the liver needs oxygen. Okay, so once the liver takes in stuff from the portal vein and it takes in stuff from the hepatic artery, it does a lot of cool magical things, and then it's going to send the blood out through the hepatic vein. That, of course, is then going to start heading to the heart, right? So what's this blood going to be like in the hepatic vein? Well, it's coming from the liver, right? And it is deoxygenated. So any oxygenated blood that was here in the hepatic artery, that oxygen has then been consumed by the cells of the liver. So deoxygenated, it's being carried at a low pressure. I know that because it's in a vein. And here, what's really important to note is that the nutrient levels have been stabilized, okay? So what the liver is doing, now my drawing looks all wonky, what the liver is doing is it's getting stuff that's coming in at a variety of nutrient contents, right? But whatever comes out of the liver always has to be steady. Okay, so what the liver is going to do is it's going to assess the blood coming in from your digestive system through the portal vein, and it's going to say, okay, do you have too much? Because if so, some of those nutrients need to come out so that this is always the same. That's an A. Okay, oh, do you have too little? Here, you can have some of my nutrients that I've been storing, because this blood always needs to come out with the same nutrient level. So what it's really doing is regulating the nutrient content of your blood. In order to perform such a crazy specialized job, we're going to need some specialized cells, okay? So these hepatocytes are just fancy pants words for liver cells. Again, hepa means liver, like hepatitis. Well, I hope you don't get that, but anyways, that's what it means, okay? And then cyte means cell, so hepatocyte literally translates into liver cell. And again, the function of the liver, which includes hepatocytes, is to either remove or add substances from the blood to maintain homeostatic levels. So everything that leaves the liver needs to be constant and regulated and within an error range. And so the liver's got a hard job because there's a lot of crazy stuff entering here. It's not always on the same levels of nutrients and such, okay? And its job is to regulate that kind of stuff. All right, so let's take a look at what exactly is going on in your liver here. I get worse and worse every time I draw this. Okay, so these two blood vessels that are entering here, this hepatic portal vein and the hepatic artery, once they get into the liver, they're going to start branching into venules and arterioles, okay? So branching smaller and smaller, okay? You get the idea here. So once they do that, these really small venules and arterioles are going to be fed through these little tube-like structures that look like this, okay? And these guys are called sinusoids. All right, so let's go ahead and label the parts of a sinusoid. This blood here, I can tell, is deoxygenated. So this is actually a really small blood vessel called a venule, and it originates from the hepatic portal vein. Okay? This really small blood vessel we call an arteriole, and this guy is from the hepatic artery. Okay, so when we think about our blood diagram that we just did a couple of minutes ago, hepatic artery bringing stuff in, okay, hepatic portal vein bringing stuff in, and that's just what we're seeing here, smaller branches, no big deal. These brown cells here that make up kind of like the walls of the sinusoid, these guys are those hepatocytes that we mentioned. Hepatocytes, okay? And these little pink guys here, these are what we call endothelial cells. So we've been talking mostly about epithelial cells. Remember, epithelial cells are those cells like kind of like these that line the villus on the outside. Epithelial, EPI, are on the outsides of things, right? endothelial cells are on the inside. Okay, so a little bit different here, but you can always remember that based on that prefix. Okay, then inside we have these weird yellow cells, these specialized cells. Okay, and these are the Kupffercells. That's 2 F's. We'll talk more about their functions in just a little bit. And so what's going to happen is blood comes in from the hepatic portal vein, from the hepatic artery. That oxygenated and deoxygenated blood is mixing. These specialized cells are all doing their liver jobs. Okay, these cells use up the oxygen that's in this blood, and then after all of the nutrients have been regulated and the oxygen has been removed, all of the blood is going to exit here through this venule. So again, another small vein. But this time it's going to end up joining to the hepatic vein. Okay, so remember that's the one blood vessel, the hepatic vein, that is leaving the liver and headed towards the heart. Now, sinusoids aren't the only teeny tiny little tubey blood thing that we've talked about, right? So we've talked quite extensively about capillaries. So capillaries and sinusoids both are very teeny tiny, very small, skinny blood vessels, but they differ in a couple of ways. Sinusoids are just a tad bit wider, okay? So capillaries really are only large enough in diameter to fit one red blood cell through. Okay, those sinusoids are going to be a tad bit wider in diameter. Sinusoids, as you might have noticed, are also lined by those endothelial cells. They were pink in that diagram we just looked at. And you may have noticed that they have gaps between them. Well, those gaps are there on purpose. And so what those gaps are going to do is they're going to allow for stuff to pass through. So we call them fenestrations, okay? And what that's going to do is it's going to allow stuff that's in the middle of the sinusoid to kind of get out and go through that hole and return into the bloodstream. So it's kind of like a filter. These gaps act as a filter. Okay, sinusoids also contain these things called Kupffercells, and they break down old hemoglobin from old red blood cells. We're going to talk a lot more about that process later, but when you think about recycling hemoglobin from old dead red blood cells, we need to keep these Kupffercells in mind. And another reason they're different is because sinusoids are receiving a mixture of oxygenated and deoxygenated blood. So capillaries kind of work like this, okay? Oxygenated blood goes in. Some of that oxygen diffuses out through the capillaries to the tissues of the body. So then once the blood leaves the capillary, it's deoxygenated. Well, these sinusoids receive both oxygenated and deoxygenated blood at the same time. Of course, it's all deoxygenated when it leaves, but this is a pretty unique scenario over here. So you can live without some things, right? Like you can live without your nose, and you could live without your hand, and you could live without your appendix, okay? But you cannot live without your liver. And one of the things that your liver is super amazing at is removing toxins. We put a whole lot of bad crap into our bodies all the time, okay? So common sources of things that are highly toxic include pesticides, preservatives, alcohol. Okay, so even if you don't drink alcohol, it's likely that your food, or at least some food that you eat, contains preservatives or maybe even trace amounts of pesticides. And our liver has to remove that stuff from our bloodstream. So we're going to use the two specialized cells in the liver to do that. Kupffercells are really good at phagocytosis. Okay, so remember, phagocytosis is a form of endocytosis, and it looks like this diagram. So here is a Kupffercell, this beige beauty here, and it is literally engulfing a toxin. Okay, so these Kupffercells are going to do that. They're also going to use that process to remove old red blood cells. We don't want them from the bloodstream. We also don't want any bacteria. Okay, so these guys are going to remove those large objects with phagocytosis. And once they're in here, once they're in the cell, we need a whole lot of lysosomes that contain enzymes to like attack and break down and kill whatever that was that it engulfed. Okay, then we've got these hepatocytes. Those were brown in our diagram. They line the sinusoid wall, or they make up the sinusoid wall. These are the most common. So these are like those tricky little sneaky guys. They don't engulf anything, okay? What they do is they chemically change the toxins to make them into something that's less dangerous and water soluble so that you can just pee it out. So I often think about these Kupffercells as being like the army of the liver. Like, I'm going to go in there and I'm going to destroy things. And meanwhile, these hepatocytes are more like the CIA. They're like a little bit more subtle. They're not destroying anything. They're just changing it so that it's not so dangerous, and then you can pee it out through your urinary system. One of those toxins that needs to be constantly regulated and dealt with by the liver is alcohol. Okay, so how does this work? Well, assuming that you drink your alcohol like normal people, okay, that alcohol is gonna come through your esophagus into your stomach, and it's gonna make it into your small intestine. From your small intestine, that alcohol is then going to leave through the hepatic portal vein, and it's going to go to the liver. Okay? The liver is going to try to do what it can to get rid of that alcohol toxin, and then that blood is going to return to the heart. Okay, I know that's not what your heart really looks like, but play along for a second. Then let's think about where blood goes from the heart. From the heart, because we need oxygenated blood, that blood is going to go to the lungs. These are lungs. You'll just have to trust me. Look, there's a trachea. Okay, it goes back to the heart, and then it goes back to the liver. So if there's any remaining alcohol in this blood that's leaving the liver the first time, it's going to come back a second time. So all blood goes through the liver twice, just like all blood goes through the heart twice. The liver is the same way. So it's going to go through right from the digestive system. It's probably not going to be able to get all the alcohol out at once, especially if you're drinking quickly or heavily. So some residual alcohol is going to be in the blood that leaves the liver. It's going to get pumped to the heart and lungs and come back to the liver again. So people with chronic alcohol abuse problems often experience various forms of liver disease. We'll talk about that later. And that's literally because it passes through twice. So guys, be nice to your liver. Take care of your bodies, okay? You only get one of them that I know of, okay? Your liver is a pretty resilient organ, okay? So it can repair itself to a certain extent, but after a while, when you keep on doing the same old stuff to it, okay, it can have some dramatic long-term effects. Okay, one of those is inflammation. So inflammation just generally of anything means swelling and discomfort. So you can have a swollen liver. You'll often see a distended, that means like swollen belly of long-term alcoholics. That's not because something is wrong with their stomach. It can be an indicator that their liver is inflamed and swollen. The other thing that can happen is called fat accumulation. So when you consistently abuse your liver with alcohol, some of those sinusoids are going to die. They just give out. They can't do it anymore. They quit. Okay? So you're going to have kind of like holes in your liver there. Those holes are going to be replaced with fat. Okay, so this is when fat takes the place of normal liver tissue. This is not a good situation here. Okay, a fatty liver is what we call it. is not a functional liver, because those sinusoids aren't just processing alcohol, they're processing nutrient contents and a whole lot of other things. So if you've killed the liver and replaced all those sinusoids with useless fat, that's now less liver tissue that you have for the liver to do its normal job. And the last one that we're going to talk about here is probably the one that you're most familiar with, which is cirrhosis. And cirrhosis is kind of like that next step in liver damage. So first it's swollen, like warning, warning, and then it starts to replace its tissue with fat. Okay, eventually when it just can't do it anymore, those healthy sinusoids are going to be replaced with scar tissue. And this is literally what a liver with cirrhosis looks like. You can imagine that this is not getting a whole lot of work done. So it's unable to do a lot of the things that your normal liver functions would include. Okay, so let's remember that the job of our friendly liver here is to make sure that whatever blood is leaving the liver has a constant homeostatic range of nutrients. So no matter what's going on in the digestive process, whether you just had 9 Shamrock shakes or whether you haven't eaten in 12 years, okay, your liver needs to make sure that that blood leaving there has a constant and regulated level of nutrients. So let's think about your digestive process, right? Let's say you're eating a lot, okay? That means the blood going to your liver is going to have way too many nutrients. If you haven't eaten recently, okay, then that nutrient level is going to be too low. So what the liver does here is it will either add nutrients to the blood or take nutrients out of the blood to make sure that no matter what crazy crap it's getting, that the blood leaving there is always good to go. Let's take a look at glucose for an example, our favorite molecule, glucose. Okay, so after a meal, okay, my blood glucose levels are likely to be pretty high, okay? So if I just ate a meal, I've got a high glucose level in my blood. So the first thing that's going to happen is that the hepatocytes, those liver cells, are going to absorb the glucose. They're going to take it out of the blood. There's too much in there. We can't let that leave the liver. They are going to convert that glucose into glycogen. So remember, I can take glucose monomers and join them together by an anabolic condensation reaction. I can join a bunch of them together to make a huge molecule called glycogen. When I do that, that's kind of cool because that means I'm removing it from the bloodstream. Okay, so I'm taking the glucose out of the bloodstream and I'm storing it as glycogen in those hepatocytes. So they have the ability to store that stuff. When you look at an electron micrograph of a liver cell, it's a dead giveaway. If it's a liver cell, it's going to have some mitochondria, it's going to have a whole bunch of lysosomes, and it's going to have these weird black polka dots that are too big to be ribosomes. Well, those are actually glycogen, okay? So each of these tiny green things is a glucose molecule. When I string a bunch of them together, I can get a glycogen granule that I can actually see under an electron microscope. All right, so we know that the liver takes the glucose out of the bloodstream and turns it into glycogen when the blood sugar levels are high. But what about like when they're low? So I haven't eaten all night, I'm waking up in the morning, and I haven't eaten for like 8 hours. What is going on with my liver? Well, the liver is kind of freaking out. It's saying, holy crap, this blood I'm getting from the digestive system has nothing in it. This really sucks. I can't let this blood leave my liver without having anything in it. So those hepatocytes are going to take some of the glycogen that they stored from earlier and they're going to break it up. Okay, so they're going to send in some enzymes to break this up into its individual glucose monomers. So it's just going to break all these bonds via a catabolic hydrolysis reaction, okay, and it's going to release those glucoses into the bloodstream. So the hepatocytes are taking out glucose after you've eaten because there's too much and putting glucose in there when you've been fasting for a long period of time. All right, so in addition to storing glycogen, which we already know about, okay, the liver is a good bank or a good sink for a whole ton of other things, okay, including iron. So iron, like this kind of iron, okay, can be removed from hemoglobin molecules and sent to bone marrow where we can then make new hemoglobins. So it's a little bit of like a junkyard for old hemoglobin parts, and it's good at storing that iron. It's a good storage bank for vitamin A and for vitamin D. Okay, so vitamin D is one of our fat-soluble vitamins, and it can hang out here in the fatty parts of the liver. So when my granny was like still around, she used to eat liver and onions every Wednesday and used to gross me out. But when you think about how many nice, good sources of nutrients I'm going to find like within that liver, it makes a lot more sense. Now, speaking of iron, we know iron comes from hemoglobin, right? And hemoglobin is going to be found within the erythrocytes, or red blood cells. Now, these suckers are super important, but they don't live very long. They only live about 120 days. So we're looking at a little over 4 months. At the end of their life, if their membrane becomes weak and ruptured, it can then release the contents of those cells, which is mostly made out of hemoglobin. So what happens to all that hemoglobin? Well, we start with our friends, the Kupffercells. So those Kupffercells are going to ingest the hemoglobin using a type of endocytosis, again called phagocytosis. And they're going to recycle the parts each in their own way, okay? So remember, our hemoglobin molecules have a few different parts to them. So they have the 1, 2, 3, 4 polypeptides, and of course polypeptides are made out of strings of amino acids. They have this heme group. There's 1, 2, 3, 4. Okay, so the heme group. And then inside each of the heme groups is 1, 2, 3, 4 total iron atoms. And so those Kupffercells, after they ingest the hemoglobin molecule, are going to recycle those items in different ways. So it's going to take those 4 polypeptides and it's going to hydrolyze them into their amino acids. So here's an example of a polypeptide. Okay. If that was a polypeptide that made up part of hemoglobins, then those Kupffercells would then cut the bonds here, hydrolyze them, and then store these amino acids so that next time I need to make a protein, I have plenty on hand. All right, so that's the polypeptide. The iron is going to get sent to the bone marrow. That should say marrow. It's going to get sent to the bone marrow. The bone marrow is where we're making new red blood cells, and so they're going to need new hemoglobin molecules. So we're going to need some iron for that, and we're going to send that iron into that bone marrow. And then finally, the only other thing that was present in the hemoglobin molecules that we haven't recycled yet are the actual heme groups. And the heme groups get turned into something called bilirubin. Again, put this on your list of things. Please don't name your children, okay? Bilirubin comes from the recycling of the heme groups from hemoglobin, and it's one of the components of bile. So we know bile gets squirted into the small intestine from the common bile duct, which the liver and gallbladder share with the pancreas, okay? But that bile originates in the liver, and one of the things that it's going to need to make bile is the bilirubin. So who cares about bile? Okay, well I do, because I want to go have some French fries, and French fries have fats in them, and fats are not water soluble. So as they're traveling through your stomach, they can tend to clump together. If I have a big clump of lipid, then there's only so many spaces for enzymes like lipase to attack them. Their surface area is relatively small if it's all bunched together. So what we need to have happen is something called emulsification. And when you emulsify something, what you're doing is breaking them into small parts and then surrounding them with something that makes it impossible for them to get back together again. Okay, so it keeps them into small parts. Now, the job of emulsification like this, okay, this really important job which increases the surface area for enzymes to digest them, that's the job done by bile. So how do we get that bile? Well, we're going to take the bilirubin that we got from the recycling of the heme group, and we're going to mix it with something called bile salt. Bile salt. Bile salt is a product of extra cholesterol, okay? So we're talking about something here that's only happening in animals. Remember, plants don't have cholesterol, okay? So extra cholesterol gets turned into bile. Recycled hemoglobin or heme groups get turned into bilirubin. So I can take these two recycled things, the bile salt and the bilirubin, put them together, and that's how I get bile. And it's again going to help me emulsify these fats, break them down into small pieces so that I can have an increased surface area for digestion by enzymes. All right, I keep telling you, your liver is a multitasker. Okay, those hepatocytes that we talked about are going to help us produce several important compounds. So again, bile being one of them. The other two are really interesting proteins that kind of hang out in our blood plasma. So remember, our blood plasma, it surrounds our blood cells. Okay, it's the fluid portion of our blood, and it's made primarily out of water, but it's got a lot of dissolved stuff in it. One of those dissolved stuffs is this protein called fibrinogen. And it's going to assist with blood clotting. So all these little like net-like looking things, those are molecules of the protein called fibrinogen, and it helps to like net all these blood cells together to form a clot. And then those hepatocytes are also going to produce albumin. Albumin is something that's in your blood to help regulate osmotic pressure, to make sure that the right, like I said, osmotic pressure is being placed upon your cells and your blood vessel walls, okay? So that's albumin, also a protein found in the blood plasma, and all of these are produced by the hepatocytes of the liver. Any protein produced by the liver, albumin, fibrinogen, doesn't matter, okay? All of those things are going to be produced by the same steps in protein production that we find in normal cells. Sorry for the noise. We're having a little episode of Janitors Gone Wild out here in the hallway. All right. So these proteins that are made by the liver, same as everywhere else, right? So we're first going to take DNA and we're going to use it as a template to make mRNA. That's called transcription. It happens in the nucleus. So here's my DNA. Shouldn't have picked green, but oh well. I'm using it as a template to make a strand of mRNA with these complementary bases on it. And that's all done by the enzyme RNA polymerase. RNA, mRNA to be specific, is going to exit the nucleus through the nuclear pore. Then it is going to attach to a ribosome, most likely on the rough ER, because remember, these liver cells are producing proteins that they're going to export to other areas of the body, and we always use the ribosomes that are on the rough ER if we're going to export that protein. The mRNA is going to be translated, that's this process, and result in a polypeptide chain. So that's going to involve tRNA, anticodons, complementary codons, okay, attaching all these amino acids, etc. The Golgi is going to take that protein and it's going to further modify it and package it in a vesicle. That vesicle is going to fuse with the cell membrane. That's called exocytosis. And now that protein is out and about. Kind of like this janitor that keeps coming by here. Oh no, let's say my liver isn't working, okay? I'm having some problems. Well, a malfunctioning liver can cause a whole lot of symptoms, okay? But one of the problems with our liver is what we call jaundice, okay? And so jaundice comes from having too much bilirubin in the bloodstream. Since bilirubin is kind of a yellowish pigment, people that have too much bilirubin in their bloodstream or jaundice start to turn yellow, like that's legit, or have yellow eyes. That doesn't mean, I've always heard that like having yellow in your eyes means you've been holding in your urine for too long. That's some bogus baloney. Okay, it's jaundice, it's bilirubin from your liver building up in your bloodstream. Okay, so we've got two types of jaundice. Well, they're really the same thing, but their causes can be quite different. So infantile jaundice is typically found in newborns, okay, and they're mostly newborns that are born premature. So our liver is one of those later organs to mature. And if it isn't mature enough when the baby is born, it's not able to turn all that bilirubin into bile. So that process isn't happening. So the bilirubin builds up in the bloodstream and the baby is yellow. What do I do with a yellow baby? Well, you stick them under this blue light. So that blue wavelength of light, that blue light is going to cause some changes, molecular changes in that bilirubin, and it breaks down the bilirubin so that it can be eliminated from the body in terms of like passing through the baby's urine and feces. If you leave a baby with jaundice untreated for a long period of time, that baby could develop brain issues, okay? So it can cause brain damage if the bilirubin builds up in the brain, and it really doesn't take that long. So if you have a newborn and they don't look right color-wise, it's important that they get treated promptly. Again, this is not UV light here, it's an actual blue light, and it literally changes the molecular structure of that bilirubin pigment. Adult jaundice is the same exact thing, okay? So again, it's caused by our liver being unable to turn bilirubin into bile. That's not happening. So we get a buildup of bilirubin in the blood. Same thing, it can still cause brain problems. Okay, but here the problem isn't that you were born premature, okay? Here the problem is because you've done something to make your liver nuclear, okay? You've done something to make your liver upset, and now your liver is not working anymore. So guys, the lesson of the day is your liver is awesome. It does some really cool things. How about we take care of it, and that way we don't have to do any of this weird blue sunbathing situation. And that'll do it for chapter D3 on the liver.